right, so I'm going to tell you about sort of my experience from my original interest in trying to understand genetic causes of heart defects in general and how I've focused on Jacobson syndrome, if it will work. So my original interest, which is still an interest of mine, is in this heart defect called hypoplastic left heart syndrome. Some of you I know are very familiar with this and could probably give a lecture yourselves on it because of your personal experiences with it. So what you see here, just to tell you what hypoplastic left heart syndrome is on the left is a normal heart. And what happens is with hypoplastic left heart syndrome, you can see that the left ventricle, which is LV, the main pumping chamber of the heart, and the other structures on that side of the heart, the valves and the aorta, are underdeveloped. And so because of that, it doesn't have the capability to pump blood out to the body. And so historically, that had been a uniformly fatal heart defect. So in the womb, babies can survive because they have something up there called the PDA, which allows the heart to bypass, the right side of the heart actually bypasses the left side of the heart. So in the womb, the right side of the heart is actually what pumps blood out to the body. But once the baby is born with hypoplastic left heart syndrome, we just had this happen last week when I was on service. On a Saturday evening, a kid came in shock, dying because the PDA, that extra vessel, was starting to close. And so when that happens, there's no way for the heart to be able to pump blood out to the body. And so this is really an overall defect in the development of the left side of the heart. And so these days, as some of you know very well, there are potentially two surgical options. One is a heart transplant, but that's not a very viable option because only about 100 donor hearts are available a year for a baby, and there are somewhere between 1,000 and 2,000 babies born a year with hypoplastic left heart. So, you know, that's not a very realistic approach. So the approach that we use now is a three-stage surgical approach where we actually reconstruct the right side of the heart to do the work that the left side of the heart was supposed to do. And so many of you are familiar with that. It's called the Norwood-Glenn-Fontan. And what you're left with is essentially a two-chamber heart that then allows the heart to function kind of like a amphibian heart to pump blood out to the body. So you've heard me tell this story, but I'll tell it again. This was the first child I ever took care of with Jacobson syndrome in July of 1995 when I had just started my training in cardiology. And he has, to this day, kind of a milder version of hypoplastic left heart syndrome. His ventricle is pretty well developed, but the other left-sided structures are underdeveloped. And as I told you before, in his chart was the paper by Dr. Jacobson describing Jacobson syndrome. And this is just a little cartoon, which I wouldn't even show in most cases because this is almost an obsolete technology now, but that's obviously the cause of Jacobson syndrome. And so right away, this was really interesting to me because people can spend years just trying to find a specific chromosomal region that's associated with any kind of a heart defect or any disease phenotype. And I thought this was actually kind of you could say, a gift from Mother Nature in the sense that it already established an association between a specific genetic locus and, in this case, hypoplastic left heart syndrome. And so I spent a number of years, and to this day, still studying everything I could learn. As you saw this morning, we're still learning things about Jacobson syndrome. Let me make sure this isn't some crisis. No. So what I learned over the years is a number of really fascinating things from sort of a scientific standpoint about Jacobson syndrome and heart defects. So first of all, about half of all children with Jacobson syndrome have a heart defect, and it's independent of this. Basically, it's independent of the size of the deletion. So whether it's a small deletion or a large deletion, you can still have a heart defect, and it can be any number of heart defects. And you can actually also have two children with the identical deletion size. One may have hypoplastic left heart syndrome and the other child may have a normal heart. And to me that's very fascinating because even though it says that there must be something in 11q that's responsible for causing heart defects, but then there must be some other factors that can somehow override that to, in some cases, allow for the heart to still develop normally. And so of those half or so of the kids that have heart defects, I've kind of broken it down into two categories. So the first category, which accounts for about 2/3, are those that have basically anything either affecting the left-sided structures or a hole in the heart called the ventricular septal defect. And one thing that was actually astounding was, as evidenced by the group here, is that about 5 to 10% of your children with Jacobson syndrome are actually born with hypoplastic left heart syndrome. So that's a couple thousand times more common than the general population. So obviously there's something important in chromosome 11 that must be at least predisposing to hypoplastic left heart. And so in perspective, that probably means that about 1 to 2% of all the patients born with hypoplastic left heart syndrome have Jacobson syndrome. Just out of curiosity, let me show you the next slide, because the other remarkable aspect from a scientific standpoint is that the vast majority of the other major heart defects that occur in the 1% of the general population with congenital heart disease also occur in Jacobson syndrome. So I'm just going to take advantage of the fact that I have you in the room. I just want to hear from all of you. First of all, how many of your children have a heart defect? Wow, okay. Let's go around the room. Some of them I know, some of them I don't, as far as what kind of heart defect. If you guys are comfortable doing that, is that okay? If you don't want to say it, you don't have to say it. VSD, VSD.
VSD and ASD.
Aortic valve stenosis.
VSD. ASD, VSD. Did I? ASD. VSD. Total anomalous. Go ahead. It's 5 things.
ASD, VSD, TAQVR, and cord triatriatic.
And there were a few hands. Hypoplastic left heart.
Hypoplastic and VSD.
Mother-son, excuse me. Okay, so pretty well represented, but I think suffice it to say hypoplastic left heart and VSDs are among the most common of all comers. And so, so already I think that made this a very interesting syndrome to try to pursue in terms of understanding the biology of the cause of these heart defects. So again, about half of your children have heart defects. There's an unprecedented high frequency of hypoplastic left heart syndrome compared to the general population. There are a lot of genetic syndromes with with heart defects, but nothing compares amazingly to Jacobson syndrome in terms of the frequency of hypoplastic left heart. So here I was, completely ignorant, naive, wondering how can I possibly even begin to study hypoplastic left heart from the genetic standpoint, and in my first month of cardiology training I'm already exposed to this opportunity. And as I said, the majority of, let's say, the heart defects of the children like I just took care of last week on service occur in Jacobson syndrome. And so I think, again, it's an extraordinary opportunity to allow us to learn not only about the basis of the heart defects in your children, but presumably how that applies to the general population. And so we also knew, as I think all of you appreciate, that from the earliest reports after 1973, that the deletion sizes are not the same. And so the strategy was, in essence, to try to find the smallest region that is deleted in a patient with Jacobson syndrome that has a heart defect, to try to pinpoint where the gene is. And then from that region, using now some of the really exciting molecular technologies that you've heard about, we can actually know what genes lie in that region, and then it allows us to make some educated guesses to pursue what might be the actual gene. And so I don't know if Marie showed this the other day, but this is what we refer to as the chips that we use. So on that little black square can be as many as a couple million little dots, and each one of those dots represents a part of every chromosome that allows us to determine whether or not a region is deleted or duplicated. And so over the years, I've come across a number of patients, and a few in particular that have helped us to really refine that region that, when I first started, contained about 200 genes, down to a region that might contain only 5 genes or maybe even 2 genes. And so that's Edgar up there. The little girl up on the right was a little girl from Colorado who was born with hypoplastic left heart syndrome, who actually had a heart transplant and unfortunately passed away from complications from that about 5 years ago when she was about 10 years old. But she had a much smaller deletion than I had ever seen before in a child with hypoplastic left heart syndrome. And so that allowed us to narrow down that region from maybe 200 genes to, I can't even read this because I'm getting old here, something like 50 or 80 genes. And then quite amazingly, thanks to this new technology, there was a baby born in New York about 4 or 5 years ago with hypoplastic left heart syndrome that had a deletion in chromosome 11 and 11q, but it didn't go out to the end. And that's what we call, as you've heard, an interstitial deletion. But it turned out that deletion, as well as the deletion of Christian, who has an atrial septal defect, turned out to overlap, as I'll show you on the next slide, the deletion of the little girl that had the heart transplant and passed away. So what I'm trying to tell you in words is what I show up here. So if you look at that black line, that's the end of chromosome 11, and the blue line is that region of the little girl that I just showed you that contains about maybe 80 genes. It's still pretty challenging, although remarkable when you think about it, that you can go from 30,000 genes down to a region of 80 genes. But that's still pretty difficult to try to figure out which one of the genes out of 80 is the cause. But again, as I mentioned, what was remarkable is we came across a small number of very interesting patients that essentially have the features of Jacobson syndrome, including the heart defects, but have these interstitial deletions. And what it allowed us to then do was look at the region of overlap. So now we were able to bring it down to a very small region that contains only 5 genes. And I'm not going to go through all this except to tell you that thanks to some other published reports, it suggested that one of these 5 genes was very likely to be the gene that at least causes some of the heart defects. And it's a gene called ETS1, or ETS1 as I refer to it. And I just want to tell you a little bit about ETS1. So ETS1, actually I'm very interested in talking to Ken Paris about this because ETS1 was originally found in white cells and lymphocytes. And it turns out that ETS1 for a long time had been known to be critical for normal white blood cell development. So I actually think this may be a very relevant gene that could be contributing to the immune deficiency problems that you heard about this morning. But actually, nobody knew anything about the possible role of this gene in causing heart defects in humans or even mammals. So what is ETS1? ETS1 is a part of a member of a family of proteins or genes called transcription factors. And transcription factors are a very complex group of genes that regulate pathways in the development of a lot of different processes, including organ development. And I'll give you a little more description about that in a moment. So interestingly, it turns out, to our satisfaction actually, that if you look at the small number of genes that have been shown to cause human heart defects, most of them indeed are transcription factors. And so the question is, how do we now try to study this gene to, first of all, prove that it really is causing heart defects, and then to try to understand what its actual function is to maybe then use that information to come up with ways to treat better or ultimately prevent heart defects. So don't worry about this slide. I know everyone's brains are tired from this morning's sessions, but one of the things that you want to be able to figure out, though, is where is this gene functioning with respect to heart development. And so the heart, as you can imagine, like all organs, is a very complex group of cells that have to perform or develop precisely. And so we refer to that as different cell lineages. And it turns out all this picture is, all those colors are actually showing you where the ETS1 protein that the gene encodes is actually functioning. And what we were able to show then is that these are cross sections actually through a mouse heart, that the ETS1 protein is actually expressed during heart development in 2 lineages or cell populations that are critical for heart development. One is called the neural crest, and the other one is called the endocardium. And I'm going to spend a lot of time, the rest of my time, mostly talking about what we're beginning to think the ETS1 protein is doing in that population called the neural crest. But before we can do that, this is still sort of circumstantial evidence, and we're trying to build our case to really prove that ETS1 really is the gene. And so, as you've heard earlier, we can actually specifically eliminate a gene from mice. And by the way, it's a little bit humbling to realize that the difference between a mouse and a human is really not that much. We share about 80% of the genes or so. And so it turns out mice have this same gene. And yeah, we're really not that different. And so what we could do then is specifically delete or get rid of that gene in the entire mouse genome. And so what you see there on the left is just a normal mouse heart. And I think you can kind of appreciate that these two chambers here are the ventricles, and in this angle you see the main blood vessel called the aorta that comes out to supply blood to the body, and then the two upper chambers are the atria, okay? And then when we eliminate the ETS1 gene, what I hope you can see here is that this wall, which normally is intact and separates the two ventricles, has a big hole in that. So anybody know what that is when you have a hole in that wall? The most common heart defect I heard you guys say, a VSD. So there we go. So now we've been able to show that at least in this animal model, starting with the human genetics approach, we think we found the gene that causes the same heart defect that many of your children have that you told me today. In addition, and that happens at least in one specific strain of mice in virtually every mouse we look at. What is also interesting is if you look at the heart on the far right, if you compare it to the other two hearts, you can see that this wall down here is very thickened, and actually this chamber is actually kind of smallish, the left ventricular chamber. So that volume is small. And remember, I showed you that picture of what hypoplastic left heart is. So it looks like in a subset of these same mice that have a ventricular septal defect, their left ventricular chamber is underdeveloped, mimicking or reminiscent of hypoplastic left heart syndrome in humans. So I think that provided some compelling evidence to allow us to argue that ETS1 is causing at least some of the heart defects, at least VSDs, and maybe a hypoplastic left ventricle. So now I want to come back and tell you about this interesting population of cells called the neural crest, or the cardiac neural crest. And it turns out when the baby is developing in the womb, a population of cells that are required to form some of the structures of the heart actually originate in the developing central nervous system. And so if you look at a developing embryo, there's what's called the neural tube, and a part of that tube is actually ultimately fated to be, to become parts of the heart. And what happens is that those cells, called the cardiac neural crest cells, have to, during critical times of development, migrate from the central nervous system to the developing heart to the exact right places to then form those final structures, okay? So it has to be a very precisely regulated mechanism both in time and space, all right? And again, we showed that the ETS1 protein is expressed in these cells during the earliest times of heart development. So that raises the question then, what is ETS1 doing with respect to heart development in this population of cells? And I'll come back to that in a moment. So one of the things that I mentioned already, though, what I think is very interesting is you already heard that you can have children with the same deletion and either they have no heart defect or potentially a variety of different heart defects. And so what we think that's due to is the other genetic differences between individuals. So we call that like a genetic background effect. And so recently we've actually deleted the ETS1 gene in another genetic strain of mice, and very interestingly we're seeing some other heart defects now. So I think you can appreciate here that's not subtle. So you can see the left ventricle of this heart, and look at the right ventricle, it's almost completely obliterated. So that's called basically a hypoplastic right ventricle or hypoplastic right heart. So it's beginning to give us clues that are consistent with what we already knew in the human situation, is that whether or not you get the heart defect and what type of heart defect depends upon the genetic background. And that's really important because then it allows us not only to identify or have an opportunity to study how these different heart defects develop as we see more of them, but I think the ultimate question that I'm interested in is what are those genetic factors that determine whether or not you get a heart defect or what kind of heart defect. And here's another example of now what happens in a different genetic background. This actually is a little bit hard to tell, but there is a hole here, a ventricular septal defect, but now the aorta is kind of pushed over to overlap partially over the right ventricle as well, and so we call that an overriding aorta. And it turns out that in this heart, here's the aorta, there's almost no pulmonary artery. The pulmonary artery is the artery that supplies blood from the right side of the heart to the lungs to get oxygen to the blood, and then that oxygenated blood comes back. So now here's another major heart defect that we see, again, in this new genetic background. So again, I think it points to Now, the fact that we have an animal model that's going to allow us potentially to study these differences in the cardiac phenotype. So I told you that nothing was known about ETS1 in mammalian heart development, but if you join me this Sunday to go for a swim in La Jolla Cove, if you look carefully, you'll see these little organisms growing on the side of docks or on the side of rocks. And this is something called a sea squirt. So a sea squirt is just a tube. It's actually, I'm not sure if it's even considered to be an animal, but it actually has a heart. And it comes in all kind of shapes and beautiful colors. And it turns out a colleague of mine who is in Arizona, University of Arizona, who's a PhD researcher in cardiac development, has spent his whole career studying heart development in this sea squirt. And let me show you what a heart in a sea squirt looks like. This is a picture from a textbook that was published in 1900. And it turns out the sea squirt has this tube that actually functions as a heart to pump its version of blood out that tube to the rest of the body. And let's see if this works. This is actually, if I can get it to work, the beating heart of a sea squirt. Isn't that cool? I got to do it again. I just think it's just so cool. As my son would say, do it again. All right, so that's a sea squirt's beating heart. And even though, like I said, nothing is known about really the role of this gene in mammalian heart development, but it turns out this gene has been shown to have a critical role in the development of that heart in the sea squirt. So yes, we're pretty similar to a little organism in the ocean. It's a little squirt, right. And what I'm showing you here is actually something amazing. If you see these two cells right here, these are the first two cells in the sea squirt that end up developing to become that heart structure. These other two red cells actually end up to develop another type of muscle, just the tail muscle of that little thing. And unfortunately, since I just recently upgraded my software, I don't think it's going to allow me to play this, but these green cells actually are the cells of the heart in the sea squirt. Yeah, it's not going to work. But what I was hoping to show you is that these two cells on the left have to move to the right location to become the heart, and they have to somehow know that I'm designated to become a heart. So it turns out ETS1 in the sea squirt has been shown to have a crucial role in regulating those two critical developmental processes in a sea squirt, namely heart cell migration and the ability to actually differentiate into a beating heart. All right, so coming back to now the human or mammalian model, this is actually a colleague of mine who's based in Chicago who is an adult cardiologist who kind of stumbled upon the same gene for a very different interest, but he did something that's very interesting. What we're seeing here is on the left, those blue dots are actually neural crest cells that are making it to one of the parts of the heart that goes on to form the ventricular septum, suggesting that normally those neural crest cells actually are critical for formation of that septum. And if you look at the same picture in a mouse that's lacking the ETS1 gene, you don't see any blue dots, okay? And so what that suggests is the loss of ETS1 is somehow affecting the ability of these neural crest cells to get to that final structure to form that structure in the heart. We don't know what the mechanism is. So that's in a fairly mature heart, and we've gone on to begin to look at earlier stages of heart development in the mouse, and I'm not going to walk you through this. Well, I guess I can. I can just show you real quick. This blue streak right here is a normal heart, showing that streak as the heart cell is migrating to the developing heart. And you can see it on this side as well, just a left side versus a right side. And notice here's the head up here. You see that red dot? Okay, there's very little blue right there. Okay, so that's the facial structures of the developing mouse. Now, if you look down here at the same stage mouse that has had the ETS1 gene deleted, I think you can see that compared to that big blue streak, there's a lot less blue streak. And we think what that's indicating is that there are fewer cells getting to that final structure in the heart. But in parallel to that, what you also see is look at the face. See how the normal mouse, there's no blue staining? I hope you can see this, that in this mouse now, where there are less neural crest cells going to the heart, now it looks like there are neural crest cells that are making it to the face. So that is a very intriguing observation that we still have a lot of work to prove, but it suggests that maybe the effect of ETS1 is causing sort of a trafficking problem, so that cells that are supposed to go to form the heart are now making a wrong choice and going to the face instead. And so we're actually doing some comprehensive three-dimensional imaging using CAT scans to analyze these mouse hearts and face to see if there's any difference in the facial features, as well as making three-dimensional reconstructions of the heart. And this is just a quantitative way of showing the same thing. So at the relatively early stage, there's more signal going to the head when you lose ETS1 compared to the normal. And then a day later, now there's not much difference between the head, but now there's much less signal going to the heart or the body rather than in the normal. So that's in mouse. And we can actually use a similar kind of genetic trickery that's actually, again, easier to study the role of ETS1 in heart development in chicks, actually. So now we're bringing chicks into the picture. And this is just to show you that you can actually specifically shut down the expression of the gene of ETS1. And what you see here is this is one side of the embryo compared to the normal side. And when you basically inactivate ETS1 on this one side of the heart, of the embryo, you see that there are also less neural crest cells on that side, namely less blue streak than on the non-injected side, or the control side. And we've gone on now to ask the hard question in chick, what happens if you just turn off ETS1 in neural crest cells rather than in the whole animal, which is what we originally did in the mouse. And this is a preliminary result, but what this arrowhead shows you is in a chick and where ETS1 was completely turned off just in the neural crest, we get a ventricular septal defect. And this is just the control on the left. And so, again, that takes us back to the fundamental question, what's going on in this population of cells? Somehow, if you look way down here, there's a very small population of these neural crest cells that go right here, and that's where the septum is that's lost in your children that have ventricular septal defects. So we think perhaps that somewhere along this whole pathway, that process is messed up as a result of losing ETS1. So now that's interesting. We've still got a lot of work to do to prove that, and we're actually in the process of doing a similar experiment in the mouse that I just showed you in the chick, where we can specifically inactivate the ETS1 gene in the neural crest in the developing mouse. This is a collaborator of mine from Caltech. You're expected to memorize this whole slide by day's end. All this is meant to tell you, though, is that ETS1, I can't even see it, it's right there. ETS1, remember I told you it's a transcription factor? What do transcription factors do? They regulate pathways of genes that are critical for developmental processes. And this is what's known already about ETS1 in neural crest cell development in the chick. So you can see why we have an awful lot of work to do, because what we really, at the end of the day, want to do is figure out what are those genes that ETS1 is regulating, because maybe there would be a way to replace the function of some of these genes that are downstream of ETS1, the so-called targets. And more importantly, maybe there are ways that we can actually preserve neural crest cell function. I'll give you, I'm just going to go off on a tangent for a moment, but I want to give you a little example. Most of you that have been pregnant probably took folate. Do you guys, anybody know why you took folate when you were pregnant? Because your doctor told you to, right? Folate is actually thought to prevent the development of neural tube defects, okay? Folate is a vitamin, and it's thought that actually the way by which folate prevents neural tube defects is to preserve neural crest cell function. Actually, it protects or stimulates neural crest cell proliferation. So it's thought that if you are deficient in folate, you might have, sounding like a familiar theme, decreased neural crest cells that lead to the development of a neural tube in some cases. And I think what we're starting to see with the ETS1 story is that if you have something that decreases the numbers of neural crest cells that are meant to go to the heart, you get heart defects. So my ultimate dream and goal is to maybe find whatever these factors are by doing this kind of extremely tedious work that might allow us to come up with something like folate that could help us to prevent these heart defects from ever occurring in the first place, almost regardless of what the genetic cause is, meaning whether you were going to have Jacobson syndrome or maybe some other gene or maybe some other environmental factor that could, again, affect neurocrest cell development during that critical window of time when the heart is developing. And so just to summarize, the majority of the most common and severe congenital heart defects, including hypoplastic left heart, occur in Jacobson syndrome. Thanks to these recent technologies, we were able to go from the whole genome of 30,000 genes down to a region of 2 genes. And these days, actually, we could do this in a very short period of time if you happen to come across the right patients. And most importantly, that loss of ETS1, at least in mice, causes some, maybe eventually many, if not all, of the same heart defects that your children have, but in a very genetic background specific manner. And so I've kind of said this 5 times already, but our hypothesis then is that the loss of this gene causes congenital heart defects because it somehow affects neurocrest cells from being able to get to the heart, maybe because of some combination of their inability to migrate there. Maybe they die off for some reason before they can get to the heart, or maybe they get there and somehow just don't have the, or they get close to there, but they're not really differentiating into heart cells. Instead, maybe they go to the wrong place and then they develop into the facial structures. So I haven't talked to you today about our other ideas that are not that far along as far as the role of ETS1 in at least one of the other cell lineages that we have shown it to be expressed in, namely the endocardium. But I suspect, for example, total anomalous pulmonary veins is probably not a neurocrest cell defect, but it's probably more an endothelial cell defect, and so that's something else that we're going to be looking very closely at. So future studies, I already told you we want to do that heart experiment, which is in the works, to delete ETS1 specifically in that population of cells in the mouse. And again, we have a very powerful opportunity to use the mice to determine or to identify what that factor or factors might be that determine whether or not the mice have a heart defect. And I think you can appreciate the important relevance of that to human disease. And finally, in case all of you were wondering why was it I wanted to put your poor kids through the torture of a skin biopsy, which hopefully wasn't too bad, we want to use this technology that is, I think, absolutely extraordinary, that's just been developed in the last few years. And I'm going to show you the same slide that I showed you yesterday, showing us that we have the ability to then to reprogram these cells to become basically any kind of cell we want, including neurocrest cells, thanks to a collaborator of mine here in San Diego. And with that, then we're going to take your children's skin cells and differentiate them into neurons in some cases, but we can also differentiate them into endothelial or endocardial cells. We can differentiate them into cardiac myocytes, the main muscle cell, and most relevant for this discussion, my colleague here in San Diego is the first one in the world to show that he can redifferentiate your children's skin cells to become neurocrest cells. And that way we can then determine in the Petri dish, whether there might be some of these abnormal properties of neurocrest cells compared to normal neurocrest cells. And with that, that actually empowers us then to maybe select for drugs or factors that could potentially restore those functions. So that's why I asked for your children's skin samples this week. All right, and as you can imagine, I'm a small player in all of this because there are a lot of people that work with me to make this a reality. I want to tell you a little bit of a humbling story about my friend and colleague, Chris Jones. I don't know if any of you know Chris. You do, from back in 1999 when Chris came out here. So shortly after I started this research, actually in 1998, when I was actually back in the lab after my clinical training, I spent 3 months living in London working 7 days a week with Chris Jones, because Chris was one of the very few people who was actually studying Jacobson syndrome. Chris is a PhD, earned his PhD at Cambridge in molecular biology, which is probably the mecca of molecular biology. If you know the story about Watson and Crick, who discovered DNA, all that happened at Cambridge. So Chris is a real talent who showed me everything that I needed to learn to be able to then come back to San Diego and start this research applied to human disease. Chris has always been challenged like I am to get grants, and finally just decided the hell with it. And so he left science altogether just a couple of months ago, and now he's starting his own microbrewery. So I'm sure he's going to make a heck of a good beer, and maybe someday we'll have that to serve here. But I think it is actually a kind of a humbling reality check that someone as bright and talented as Chris has had to give up science altogether because of the way the funding is. And so that's a good reason, example, why we're going to have that fundraising session tomorrow to kind of brainstorm. Because I think that's just a real loss to everyone. A gain for the beer drinkers, but a loss for science and humanity. And then I have a number of people in my lab over the years. Mao Ching Yi, who just recently went back to China, is a very talented MD, PhD, who did most of the work that I showed you. And then I have two new postdoctoral fellows, Yan Yin from China and Kazumi Fukatsu from Japan. Kazumi actually had been working in Marianne Bronner's lab, the one who I showed you that very complex slide up at Caltech. Leanne Garrett-Sinha actually is someone quite interesting who we should probably recontact, because she's the person who's been studying the role of ETS1 in T cell or white blood cell lymphocyte development. And so we should probably revisit with her as far as trying to reconcile her work as far as the role of ETS1 in lymphocyte development and a lot of the clinical problems that I, for the first time to a large extent, learned this morning. And of course, Teresa, I think you know how invaluable a resource she's been to the group. And finally, obviously, all of you, because none of this would be possible without your support and cooperation. So I think I am done. Oh, actually, wait. The real reason why you're here is, what the hell does this mean to your kids? Sorry about that. I'm so carried away. I'm wearing a different hat all of a sudden. All right, so coming back to reality here, that was my little 20 minutes of fantasy. Hold the applause. So what are the recommendations? So first of all, I think hopefully you'll appreciate that anybody who has a child with Jacobson syndrome that has not had a comprehensive cardiac evaluation must have one. There's no question about that, given the fact that 50% or so of children have heart defects. And so that has to include an echocardiogram. Okay, so if anybody tells you your kid's heart is fine, but they didn't have an echocardiogram, you cannot be 100% sure of that. That's number one. So if your baby's heart is fine when it's done, then what I recommend is to be followed up every 3 to 5 years just to make sure that things remain okay. Generally, if you have a normal structure heart when you're a baby, usually things are going to stay that way, but there's a small chance that some things can develop over time. So in particular, if your pediatrician hears a new murmur and they tell you, oh, it's okay, let's just follow it, you can come back and tell them, no, my kid needs to go see a cardiologist. And so you should definitely recommend that. I don't think they'll question it. Something that we need to talk about is if your child develops hypertension, and you actually prompted me to think about this, there is that defect, coarctation of the aorta, which can potentially develop over time. And the consequence of that is, if you remember what the aorta looks like, imagine if you have a kink in the garden hose. So if you have a kink in the aorta, everything above that is going to be high pressure. So I don't know if when's the last time she's had an echocardiogram? Okay, so hopefully everything is okay. Say that again, I'm sorry.
They were checking the valves and the mitral valve.
I would hope that they looked at the aorta. I would think so. Or in general, if there are any symptoms that are suspicious for a cardiac problem, have a cardiologist check it out. So now I can say thank you and answer any questions.
I'll take the wireless.
Oh, okay. Actually, I think I said it down. Thanks. Yeah.
I had a quick question. I wanted to know if you can explain a little bit more about the patient that you were aware of that actually had the transplant, and explain to me a little bit about the transplant process in kids with a genetic disorder. Like, how do you get matches?
Well, regardless of whether or not you have a genetic disorder, there's a very rigorous process that has to be performed to determine whether or not a donor is compatible. But For heart transplants, and I don't pretend to be an expert on heart transplants, that's almost like a whole other field, but you need chronic lifelong immunosuppression. So the bottom line is when a donor heart comes along, they're not going to be matched in the same way, let's say, a bone marrow donor will be matched. And so consequently, those children that receive a heart transplant are on lifelong immunosuppression, and it's fraught with problems. About 10 to 20% of children that get a heart transplant, because of the immunosuppression, actually develop certain forms of cancer like lymphomas. So the expression that we use is transplanting a child's heart cures one disease but creates another. So the 10-year-old girl that passed away from the complications of her heart transplant actually had a form of rejection that caused her coronary arteries to be obstructed, occluded. So not from high cholesterol, but from the consequences of the immune issues.
How does that kind of consequences affect the potential for the malignancies and the leukemias and the treatment concepts, like bone marrow transplants and stuff like that? Where does that put them in that bracket?
Well, again, I generally do not recommend heart transplant for my patients with hypoplastic left heart syndrome unless for some reason they're not a good candidate for the three-stage. You also have to remember that the 5-year survival at a top-notch center like ours is like 85%. Right now, about maybe 20–25% of kids that are even listed for a heart transplant die just waiting for a heart. So already the numbers are working against you. So anyway, having said that, malignancy, opportunistic infections are all big issues. And so I honestly, whether or not your child had Jacobson syndrome, it could be much more complicated when there are potential risk factors for malignancies. But I don't think in my, 12 years, I don't think I've ever recommended transplant as a first-line approach for any patient I've come across with hypoplastic left heart. Does that answer the question, kind of?
No.
Okay, answered it.
What about the treatment? What about the treatment potential? Like, should our kids develop any malignancies or leukemias? What transplant bracket are they in with these type of complications in relation to maybe bone marrow transplants, stuff like that? Where does that put them?
Yeah, now you're getting me way out of my area of comfort. So you're talking about, issues of bone marrow transplant. First of all, let's say I hope to God that never is an issue for your children, but if they are, I would really defer that to a hem-onc person. I can't give you a great answer. Oh, absolutely, we gotta. Well, if you need one, sure, but in your part of the country up north, I mean, you've got UCSF and Stanford, for starters, who are as good as any place. I hope that answered the question. Go ahead.
Okay, I have one question for Dylan's mom in Australia. He is dextrocardic and has had no issues, but wants to know if there's anything that they should keep an eye on.
So this child has, basically his heart is just on the wrong side, but you can have actually a complete mirror image where all your anatomy in your thorax and abdomen is flip-flopped, but if that's all it is, it's just a mirror image, you're fine. What we have to be on the lookout for is, I'm sure Dylan has had cardiac assessments, but not infrequently when you have dextrocardia, there can be other associated structural heart defects. So you just have to make sure there aren't any associated heart defects, but if he has otherwise a normal heart, there shouldn't be anything to be concerned about, but I would still recommend he be followed every few years by a cardiologist.
What were you saying about folate treatment during pregnancy, and you said it needed to be given during the critical time. Is it not given during the critical time?
Well, if you have to give it during the time where the action is, right? So during the first trimester, I don't know, I can't remember. I think my wife had to take it all during pregnancy. Is that usually right? But so just to give you an idea, the human heart is actually basically it starts pumping 21 days post-conception, and the full structure is essentially completed by about 60 days post-conception. So think about when you even find out that you're pregnant. I mean, so, yes, absolutely. So you should take it as soon as possible once you know you're pregnant. More questions?
I was thinking about what you were saying about the earlier lecture about, sorry, I'm forgetting his name.
Ken?
Kelly? Tom Kelly?
Oh, Tom Kelly, yeah. Okay.
What he was talking about, just in response to Stephanie's question earlier, that growth hormone can help with structures, organ growth. And I know that although Elliot's TAPVR was all repaired, the one big risk growing up is that the veins don't grow with him.
Right.
And I'm wondering if there's an applicability of growth hormone to helping to promote the growth of those veins in his heart as he grows.
The one potential concern about the effect of growth hormone on the heart is that it actually causes thickening of the heart muscle. So I think the fact that he's not had any issues with his veins becoming obstructed, if I'm remembering correctly. So that's probably nothing to worry about if he decided that he needed to be treated with growth hormone. But I think it's proven, well, for many reasons in Elliot's case, having had open heart surgery, he'll need to be followed. And he should probably get, if he were my patient, I would do an echocardiogram on him once a year. And particularly if he's on growth hormone, just to be sure. Because, you know, how many people with total anomalous veins with Jacobson syndrome have been treated with growth hormone? You know, so.
I was just wondering, my daughter has simple heart defect, so I think we're probably just supposed to go maybe like one more time next year and then once every 5 years or something. Is there, because we now know that she has Jacobson's, is there a reason to say that we should be going back more often, or should that be it?
It's never it, in my opinion. When your child has a heart defect, even if it's been repaired or closed with a catheter. Did she have a device closure of her ASD or surgery?
No, it was too high.
It was too high up. So she had, I mean, there's a subset of patients, we don't know specifically for Jacobson syndrome, but we do know there's certainly a subset of children or adults with atrial septal defects that can develop problems down the road with the electrical system of their heart that could lead to something called a heart block. So it's usually not an abrupt event, it's a progressive development, and so that's a big reason, among others, why I follow all my patients that have had an atrial septal defect closed once every year or two with at least an EKG every year or two. And if she ever had like a passing out episode or something like that, that would be very concerning, you know, that she might have a problem with her conduction tissue. It's only in a small percentage of all comers, but still, we don't exactly know who that is until we know more about the genes. Pardon me? I'm sorry, I didn't.
They should be constantly checked.
Yeah, so if your child for sure has had any kind of a major heart defect requiring surgery, they should still be followed at least every 2 years.
What if they didn't have the surgery?
I'll have to talk to you about that individually and see what the circumstances are. But still, things can change even if they haven't had surgery. So,
And
sorry.
does the Jacobson syndrome, you know, affect that anyway? You said any patient with an ASD, you would do that. Does having the Jacobson?
Well, I mean, there are a few kids, for example, I can think of one, what's her name, the adult now from Arkansas, that when she was here at the first conference in 1999, her mom said she had a heart murmur, but she actually, we found, had developed significant obstruction of her aortic valve. So that's a case in point of something that can develop over time, with or without Jacobson syndrome. But I think in your child's case, the fact that she's had surgery and Jacobson syndrome, I would follow her every year or two.
Hi, have you had examples of children who were born with Jacobson whose hearts have been normal, who have developed heart abnormalities?
Right, so for example, a valve problem is something that potentially can develop over time. So that's why if you have an aortic valve problem, that's going to be manifested usually by a heart murmur. So certainly if your pediatrician hears a heart murmur that had never been heard before, that would absolutely be reason to refer to a cardiologist. And there are potentially some other more subtle changes that can happen, coarctation of the aorta like I talked about, or other valves. So at the very least, I think still that a Jacobson syndrome child, even with a normal heart, should be seen by a cardiologist every 3 to 5 years. I'm kind of selfish and protective of the kids here in San Diego. I try to see them every year, actually, because I like to. But,
can you explain a little bit more about your following up of the patients with the ASD and how they can develop the problems with the electrical system of the heart? Have you known any VSDs with that issue since Korea has the PAVCs all of a sudden? I mean,
okay, so yeah, so I have I'll give you a little bit of history. So for atrial septal defects, and actually for ventricular septal defects too, there are a few genes that we know that were discovered that can cause these heart defects. And it turns out those genes not only encode the structural aspects of the heart, but they're critical for the development of the electrical system of the heart. And the way that one such gene was found was that researchers in Boston had identified a number of families where children that had surgical repair of an atrial septal defect years later when they were in their 20s, all of a sudden were dropping dead, and nobody knew why. Until then they found the gene, and they determined that the gene, now remember, in those days you have your ASD repaired and your cardiologist says goodbye. So they went 20 years probably without any follow-up, and then something catastrophic happened. But it's almost always a progressive process, and that's been shown in mice. If you knock out that gene, the heart conduction tissue cells actually degenerate progressively over time. That leads then to heart block and overt symptoms like syncope, passing out, or even, God forbid, sudden death. So the problem is we don't know all the genes that cause these heart defects, so until we do and can say if it's gene X, then you're at risk. If it's gene Y, you don't. So at this point, I follow all of them for at least that reason. Okay.
How common is pulmonary hypertension in these kids?
In Jacobson syndrome, it's very unusual, but in hypoplastic left heart syndrome, more specifically, it certainly can be a problem. I know that Faith has had issues with that. Is she on sildenafil, by the way? Yeah. So what she's referring to is that sometimes the pressures in the lung arteries are elevated, and that's not a good thing in general, particularly in a single ventricle situation. So there are ways that we try to manage that. That's why Faith, you see, is on oxygen, and she's on medications to help lower those pressures. So I don't really know I don't know for Jacobson syndrome. I don't know an exact number for hypoplastic left heart, but it's certainly something we deal with.
Well, I'm the grandma here, but I'm a little concerned. She has a 9-year-old, my daughter has a 9-year-old, 11q deletion, and he has a aortic and possibly bicuspid stenosis, valve stenosis. And I noticed that when he plays hard, he has a minimum, when he is exerted, he gets tired. And he swims, he's on the karate team, and I'm just about to pass out, so I don't know if I'm being over. She's been seeing a cardiologist at Duke, and says just watch out for other signs of acute distress, but should he be doing all these things?
So that's a good question, and there are specific contraindications. If you have a certain degree of obstruction of the aortic valve, that can put you at increased risk for sudden death. It doesn't sound like he must have. But as long as he's being followed regularly with regular echocardiograms, the other thing that you could do that would be probably useful, he's 9 and he's running around playing, if you can coax him to run on a treadmill, then we can monitor his heart function during exercise, and we use that commonly to try to have some insurance and peace of mind that exercise is safe in our children with heart problems. But I'm glad to hear he's running around, and that's great. But our job is to be able to make sure that it's safe for him to do that. So we do have ways to ensure that.
I was thinking about you earlier saying that the issues where the veins don't grow with the kids? And would that present itself or show itself in a lack of circulation in certain parts of the body? Or, like the leg swellings and, poor circulation in lower extremities? Is that anything associated with the veins not, functioning or growing with the child, with them?
I mean, there are potential obstructions in the veins that can develop over time, like from clots and that kind of thing, but I would say, let's say in a Jacobson syndrome child, I think that would be very unusual, or any child that doesn't already have some cardiac issues. I think that would be very unusual. As
purple toes and stuff ongoing, that's not part of the circulation.
Well,
so like when the toes get purple and stuff like that, that's not part of a circulatory thing?
Well, it's actually probably more a neurologic thing because there are nerves. No, it's actually not a big, it's a benign finding. Okay. So my wife, for example, when she goes outside, if it's under 60 degrees, you would swear she has congenital heart disease because she turns blue. But the difference is, I'm going to assume it's probably intermittent, correct? Yeah, so if it's a cardiac cause, it's not likely to be intermittent, it's going to be persistent. So, and it's going to be all over, not just in the toes.
One of the main concerns with hypoplastic left heart is
One?
possibly needing a pacemaker later in life. Now, is that a direct result of the surgeries and all that? Is there any added risk for Jacobson with any gene deletions that you know of or anything?
Yeah, that's a great question. So, wow, that was like a planted question. Well, it turns out the gene that I was referring to that causes atrial septal defects, I and a few other people have found can cause hypoplastic left heart syndrome in some cases. I actually got a family where the mom and the son have an atrial septal defect and the daughter has hypoplastic left heart syndrome, and I discovered the gene, or I found the gene mutation in this gene I was referring to. So in that case, there's a genetic predisposition in a subset of children with hypoplastic left heart or some of these other heart defects that can predispose them. But for sure, the surgery that Faith has had, the 3 surgeries, also puts her at risk for developing problems with the conduction system. So a fair percentage, I would say 10 to 20% of kids with hypoplastic left heart syndrome may end up requiring a pacemaker. And one of the things I'm actually interested in is whether ETS1 may have some interaction with this other gene I'm telling you about, but I haven't heard of anybody actually with Jacobson syndrome having conduction system problems yet.
And then I had another question. The slide that you showed some migration to the face, and I was wondering with the ETS1, I was wondering if you had or if you had thought about getting with Peter Hammond on the ones that have heart defects and the dysformic features in the facial area.
The answer is yes, and we have. So as we speak, literally as we speak, I didn't have time, but I have sent our mouse embryos to a company in Utah to do 3D imaging, not only of their hearts, but of their craniofacial structures. And unfortunately, Peter Hammond couldn't make it. Originally, he had actually committed, and then he had a conflict. But the idea is to compare the results of the craniofacial structures in our knockout mice to Peter Hammond's results on your children and see if there might be any common, findings that would implicate ETS1 as causing maybe some of the craniofacial problems. That answers the question, right? No, it's a great question, and it's very interesting, right? So.
Okay, just to clarify, so electrical issues in the heart can be genetic as well?
Yes, absolutely.
Okay.
Okay. Is it time for beer or time for the breakout sessions or both, right? Great. Both.